survived, and I'd like to show them to you today. Well, I'm going to also make YouTube videos for my main channel. In a day or so, I'm going to start uploading, and I'm going to also put those specimens under the microscope for my main channel, and of course also some specimens that I'm not going to talk about here in the live stream. So of course you would like to know, where was I? Well, I was in two different places. I visited Paris, France, for about a week. I collected some samples there, and then later on I went to northern Germany, and in northern Germany I was on the on the Atlantic, on the North Sea, and there I also collected some samples. Okay, I'm not going to show them so much to you today, maybe one slide or so, but I'm going to make a separate stream, separate videos here. I'm just going to show you now generally the place in Paris where I collected the samples, and then I'm going to put some under the microscope. Well, this picture here, the glass pyramid, that's in Louvre. That's the Mona Lisa, the painting, the famous one, can be seen there. Now, those pictures I made several years ago, but these were the more current pictures. And I said, okay, that's a typical place in Paris, France, and I would like to collect some samples there. Of course, not such an easy thing. Where do you collect microscopy samples? But even though we had a drought there were fountains. Not all of them were active because they were saving water. And I said, okay, maybe I'm able to find a place here to collect some specimens. Where there is water, there is life. But if you look at it, it's pretty clean. Yeah, there are no algae there. I don't know if they chlorinated the water or not. So it's starting to loop again. So this was a little bit of a frustrating thing, but then I did find something here on the side of the fountain where the water was running down. I found some material there, something growing there. And this is basically where I took a sample and I put it into one of those tubes, as you can see here. I took it along. Okay, and not only that, I also want to show you something else that I've done a little bit experimentally. I'm going to show you my desk here again. I made, in the hotel, I made some permanent slides as well. And the mounting medium that I used is the glue, the Elmer's PVA glue, the water-based glue, which I talked about in the previous video. I simply took along the glue and I made directly right away also some permanent slides, so we can compare them as well. So basically number 2, that's the Louvre sample from the fountain. Okay, so what I'm going to do now is I'm going to take the tube and I'm going to make a slide here. And I know that there is a little bit of life going on here, not quite as much as I hoped, but there is a little bit something that we're able to see. Well, it's tube number 2, so this is the tube here. Okay, you can see that I already removed the cap again to allow some exchange of oxygen here. Because I just want to show you another one, one that I'm not going to put under the microscope today, but this one was a clear sample, okay? And it really started to decompose, it turned brown, lots of bacteria were growing there because I kept it capped, okay? So I would suggest that when you collect water samples, and of course you have to cap it when you transport it, but otherwise keep it as open as much as you can. And especially if the tube is thin like this here, it might be a problem for the oxygen maybe to diffuse all the way down. But then if there are green algae in here, maybe photosynthesis is able to compensate this. So okay, I'm just going to make a slide now, and then we're going to see what we're able to see. Okay, let's take a little bit some of the green stuff here as well. Allow the algae and whatever solid material can be found here to settle down a little bit. Okay. So this is the material that I took from the fountain. Okay. So it goes back into the jar over here, and cover glass, of course, and then let's have a look, and then maybe we can even compare it a little bit with the permanent slide that I made. Okay, so generally it was not a very interesting specimen. Okay, but just want to dampen your expectations a little bit here. So microscope view, I've got now the 10 times objective in place here. Let's have a look at what we're able to find here. And of course I need to focus a little bit. Yeah, and this is basically how it looks like. Okay, so I don't know if there is, yeah, not a lot can be seen, but occasionally, especially at higher magnification, I was able to see a lot of bacteria. So mostly there is decomposition going on here. Okay, so not very interesting. Yeah. And as a matter of fact, also when I made the permanent mount, yeah, it didn't look very much different here. But I have to, yeah, simply also tell you that some of the other specimens that I've got here, they are pretty good, okay? But I just wanted to start off with the maybe not so interesting one here. Ah, there is a little bit of movement over here. I don't know if you're able to see this. Okay. Okay, I don't know, right in the center. So let's go up a little bit. Oh, I've got the wrong setting here as well with the condenser, so I'm going to move this over here as well. Maybe this is a little bit better. Let's go up with the light. Yeah, here. Whatever that might. Oh, it looks a little bit like a rotifer. Okay. Yeah, but otherwise, yeah. You see that mostly decomposition was going on, so I did not find a lot of interesting things here. You see this here seems to be a little bit, you are able to see the individual plant cells, so I suppose some kind of a moss maybe that was growing there. Yeah. And this means, of course, when there are plants growing, then of course there's oxygen production, and it also provides a large surface for other microorganisms to grow on. Some filamentous algae. Okay, yeah, rotifers, yes. There's another something moving on over here. Yep. It's a little bit blurry. I actually was hoping to get a slightly better view here. Let's open this here a little bit. Close the condenser. Yeah. Here again. Yeah. So I hope you get the idea. I think maybe one of the reasons, I don't know really so much about that. Yeah, it's not the fountain. I don't know if they added chlorine to the water, and maybe that this could also have inhibited a little bit the growth of certain microorganisms. But then again, also don't forget that this sample here is around 2 weeks old, and it is completely normal that there is a progression of organisms here. Something else, yeah. So the number and type of organisms, of course, change as you with time. Yeah. Yeah, I was able to smell a little bit. It does. It was started to smell slightly sulfury, like sulfur. And this basically means that there is a little bit of. It's started to become anaerobic, lack of oxygen. So because maybe there were not enough plants there present or algae present in the first place to produce sufficient oxygen. So this was actually one of the less interesting specimens, but still we learned something here. I don't know what this round thing is right next here in the center. Yeah. Did you test it for chlorine? One of the questions is if I tested it for chlorine. No, I did not. But many of the, not many, all of the ponds that not ponds, of the fountains that I found in Paris were extremely clean. So there was hardly any material also growing on the side. Usually there where the water touches the side of the wall where with the surface, usually there's always something growing, but not with either cleaning it in a periodically, or they have added indeed some I don't know, some chemicals, maybe like chlorine, but I did not test it. It's a hypothesis, yeah. Because normally, if you have some kind of a water body, a standing water, and then and then you have a water fountain there, which also mixes oxygen into the whole, into the water, and you normally have, quite a bit of growth going on here, but really not so much here. Yeah. So this was actually, a little bit of a slight disappointment. So what I'm going to do now is I'm going to simply show you now the, here, I'm just going to hold it up here, the permanent slide that I made on the very same day that, just a few hours after collecting the sample, I took in a hotel, I put some some of the green stuff, yeah, on the slide here. I don't know if you're able to see it properly. Okay, and I made a permanent mount, and yeah. So I'm just gonna have a look at this, the permanent mount now. Let's put it here also, and you're going to see that there is not such a huge difference. Yeah. This is not the permanent mount. Of course, we do not expect to see any movement here. Yeah, this is using now the glue. There was a question here, what holiday? I was in France, so this is a sample taken from a fountain at the Louvre in Paris. If you joined in late, at the beginning, a couple of minutes ago, I actually played a video of the place where I collected this specimen. And yeah, so maybe after the live stream you can go on back. So this is also interesting here, because this here, that looks like it might be moss or something, okay? So there was indeed something growing there. Again, this here is now the permanent mount, okay? Which, yeah, of the fresh specimen. And as we can see, or I was able to see as well, maybe just like you are able to see, that the plants here seem to be pretty much more intact. Okay, showing that over the course of a week or 2 weeks, there seems to be some kind of terminal buds here where the plant is growing. But over the course of the week or so, yeah, the plants were also decomposed and broken down by bacteria and whatever is in there. Most likely bacteria, of course, because one of the problems is if bacteria starts to grow, then the bacteria will very quickly use up the oxygen in the water, yeah, and this basically will then yeah, immediately kill off yeah, any other larger microorganisms. Because bacteria being prokaryotes, some of them are able to also grow in so-called anaerobic conditions. That means in a lack of oxygen, and therefore, yeah, when bacteria starts to grow, then this basically means that the level of oxygen drops very quickly. Yeah. So you have to make sure that the number of organisms that are able to eat the bacteria, that they remain high, but they require a lot of oxygen. Yeah. Okay, another comment, pond water. Yes, no, actually the water of a fountain. Okay. And not the water itself, but I've scratched off some material from the side of the fountain. Yeah? And again, this here is now a permanent mount, and I just showed you. I'm going to go back here. Here, this here is now the fresh mount that I just made, and when you look at this, yeah, here there's still a little bit of a yeah, a plant visible. But again, not a lot of movement, so I was a little bit disappointed, okay? But still, I'm gonna show you a much better sample in a minute or so. Yeah? Again, if you kind of joined late, at the beginning, I've played a video on the place where I collected this here. Yeah? What were those red filaments in the permanent mount? That's an interesting one. Yeah, if it is, it could be, if it is indeed a moss, it could be the rhizoids. Rhizoids are root-like structures. Now I have to find it again here. Okay. Okay, I don't know what this is. Here could be some kind of dirt or so. There were some interesting red, yeah, that is the plant itself. There were some interesting, yeah, string-like, fiber-like filaments or structures. I don't know where it is now again. I thought that they could have been, yeah, if they're filament, or it could be fungi, of course, because they grow in filaments. I don't even know if it must have been. Okay, I lost. The thing is the following, because I'm not looking through the microscope right now. I'm just looking at the screen and over and trying to find the. Yeah, I don't know where they are right now. Okay. I mean, not every string-like structure has to be something from a plant. It could be, I don't know, some kind of dust contaminant is quite common. Yeah. But generally, yeah, as I already mentioned, yeah, rather disappointing specimen. But then again, that's also something that It depends what you expect. Why did I say disappointing? I mean, it's just the way it is. But of course, we want to see some spectacular movement or whatever. Yeah. But still, we learn something because, of course, nature is not everywhere the same. Yeah. And by looking at different specimens under the microscope, of course, we can see that different things start to grow in different places. I mean, here you see some of those filaments. Now, if they are indeed part of the plant, they could be rhizoids, which are root-like structures, I don't know. Or if they belong to, it could be fungi, the hyphae of fungi, which I probably don't think, because usually they're branched. And yeah. But I think these are the filament-like structures that, was referred to in the comment. Okay. So, so I'm just going to leave it at that with the Louvre. Okay, I want to move on now to the next specimen. I'm just going to go back here, and I'm going to start again by showing you a little video here. The next one is the River Seine. That's the river that flows through through Paris. Just want to show you something here. Just it's too small to see. I'm going to pause the video, but on the stairs there is a white thing here. I don't know if I'm able to point to it. It starts to disappear. Okay, but there's something white on the stairs. This is a person with a pressure cleaner cleaning the stairs. Yeah, and when I saw this, I said, "Wow, cool! There are some stairs, and evidently they're not locked off, so it's possible to walk down all the way to the river." Okay, so this kind of said, "Okay, I want to also collect a water sample from there." Yeah, and indeed, it's a different stair than what I just showed you, but there were kind of many of them. And I said, okay, the River Seine, that would be also a nice place to collect a water sample. So I simply walked that down, and I always take along this, you already know this probably, a little plastic container with those tubes. A boat happened to pass there as well. Just thought it's also quite nice. That's why I, took a video here, and this is basically the waves now of the boat. And then I said, okay, the water itself looked kind of clean. I mean, there were some toys there, obviously some plastic, but generally it was very clean. And I took a little sample and I put it in here, and look, okay, my head is now in the way, but I did not collect a lot of water. Okay, it's only about less than 2 milliliters. And I said, at the end I said, well, I should have actually taken along more water. But interestingly enough, this was actually a good thing. Oh, I forgot to loop the video. So I'm going to loop it again. And it was actually an important thing because there was so little water in there. Where is the tube? It's tube number 1. This is the one over here. Okay. Therefore, there was a lot of air in the tube. And therefore, and if you look at it, there are bubbles in here as well. So there is photosynthesis going on. Oxygen is being produced. So it stayed aerobic. So there is plenty of oxygen in here. And there is also some solid material on the bottom here. There are some algae floating around on the top. And this actually was quite a nice specimen. And I'm going to put this now also under the microscope. And I'm going to go back now to the desk again. Okay, so, and I'm going to now, I think this is the one that I used. I do not want to cross-contaminate. I don't know which one I used now for the previous one. So I'm going to simply not use it, and I'm going to take a fresh pipette, because when I go into one water sample, I do not want to transfer the microbes to another one, because, I just want to make sure that it remains authentic. Yeah, there is another comment. Let me just read this. Hi Oliver, I have a problem with copepods. Yes, these are water crustaceans. After a week or so, they start multiplying a lot. Eat everything in my samples. Do you have any idea on how to get rid of them? Ooh, how do you get rid of them? You know what I would have said? Don't feed them. But that's a stupid suggestion, because of course they will eat the food in there. One of the things that you might, I mean, if there are eggs around, then new ones will develop, but one of the things that you might want to try is maybe that you pass the water through a sieve. There are some metal sieves that are used maybe also for, I don't know, when you make teas. And because the copepods, they are actually much larger than many of the other microbes. And when you kind of pass the water through a sieve, then of course this will also retain some of the algae. yeah, the smaller microorganisms like ciliates, flagellates, and yeah, single-celled organisms, they're able to pass through. So this might be a possibility, but if there are eggs around, maybe, yeah, then the eggs are also able to pass through probably. But I would actually filter it, but not by using a paper filter, but by using some kind of a sieve, a thin sieve that you sometimes use for yeah, for tea, for example. Yeah, these metal sieves. Yeah, I would probably try that. Never tried it myself, but it's an interesting um, thing to try out. So okay, so I'm gonna go all the way down here, and then I'm gonna try to take something from the very bottom. Oh, too much. And a small drop here. Small drop. It's a huge drop. So what I'm gonna do is I'm going to take some water back up again. And put the rest back because I want to save it. And let's have a look at this. You want to? So I was checking. I've cleaned them already. So lots of green stuff here as well. Yeah, it seems to be a relatively thick sample. Yeah. So maybe there are some, I don't know, sand grains or some solid material in there which prevents the cover glass from lying flat on the slide. But that's okay, because I know that there are some worms in here as well. I hope that I'm able to see them now. And I don't want to squash them in any case. So I'm going to go microscope view, but I have to remove the other, oh, where is this? To remove the other slides here. Okay. So this is this here. So let's put the other one in now. So let's, this is now the 4 times. Okay, look. I think you already see that there is quite a bit more going on here. Yeah, let's turn up the brightness level here. Yeah, you see there are lots of sand grains here as well. Okay, so let's go up to 10 times. And this is 10 times. Okay, a nematode worm here. Okay, lots of sand grains. And how many of you know these little friends here, these worms? I've made already several videos of those. Iolosoma, they're called. Yeah, very common worms. And they're, yeah, I made already in my main channel some videos. Yeah. Iolosoma. They have oil globules. I'm gonna go up with the magnification, and I found a really high concentration of these iolosomas in the sample. Of course, it could be that they multiplied over the course of the 2 weeks. That's a rotifer again over there. Okay, here. Yeah. So because the sample, of course, is also several days old, it does not mean that this is exactly the same way that the sample looked like when I collected it. Yeah, but at least these organisms must have been present in the original sample in any case. Otherwise, yeah, they could not have multiplied if they multiplied. Yeah? So those are nematode worms. And look at these. How many of you know what these are? Those guys here, gastrotrix. They're cute fellas. I found many of those as well. Here, look, also so many gastrotrix. A few days ago, I didn't see as many. It also shows that they kind of must have reproduced. Huh? The gastrotrix. Yes. Now, if I go up with the magnification, then it's gonna be pretty difficult to chase them around. Okay, that's a little bit of a problem. Huh? Here in the sediment, a lot of these worms. Let's go over here on the other side. Again, a gastro trick here. You know what, why not go up a little bit with the magnification in any case, but it's going to be difficult to find them. Go up a little bit with the brightness. And then it's again a question of luck. Yeah. Where can I find them? Rotifer, nematode. Here, Iolosoma playing around with a nematode. This is again a rotifer. rotifers, they have a foot and they like to attach themselves to a surface. Okay, Iolosoma. So maybe I should go up, close the condenser a little bit more to get a little bit higher depth of field. Here. Yeah. Yep. Yeah, there's quite a bit of quite some stuff going on. Of course, all of the movement means lots of energy consumption. Lots of energy consumption requires oxygen. Yeah. So, and I do not see so many green algae in here, which might produce the oxygen. And I think, and that's my hypothesis, that because the amount of water was so little, this allowed enough oxygen supply from the air. Yeah. Yeah, the gastrotrix, where are they again? I was kind of hoping to get one up a slightly more closer view. Here's one. Focus. Here it is. Here's another one. Okay. Yeah. Okay, over here also, yeah, it's not disappeared. There's some flagellates as well. Those flagellates, they are single cell and they have a huge flagellum, like a huge extension, like a whip, which is moving and pulling them. A few days ago, there were more of these flagellates around. I did make a video of them. So when I make the video for the main YouTube channel, I'm going to, of course, give you a better view of those. Here again, gastrotrick. Because of the sand grains that you see, the cover glass is not lying completely flat on the slide, and therefore, of course, they're also able to move vertically. And this means that they're going in and out of focus as well. Okay, there's not some. Yeah, over here, this is a. Seems to be a dead rotifer over here in the center. Here again, the gastrotrick. Okay. Yeah, nice rotifer again. Yeah, here there's an algae, the green one. Yeah. Ah, Vorticella. This is a stalked cilia. And usually they contract quite fast. This one doesn't seem to contract. And, yeah, next to the rotifer is Vorticella. No, look, the worm. No, not the worm. The rotifer is touching the Vorticella. The Vorticella does not contract. It's an interesting thing. Nematode. You know what? But that's actually also an advantage. If it doesn't contract, then I might be able to. It's too dark. See it a little bit better here. Let's see if it's still. It's actually. Yeah. Oh, now it contracted. Okay, this was it. This is what I wanted to show you. Ah, yeah. Here at the top, do you see this? This one single cell. That's a flagellate. Look at this long thread-like extension that it has. That's the flagellum pointing forward and pulling the rest of the cell along. Yeah, some testate amoeba. I saw some amoeba tests, which are some shells. Yes, I have seen them, but not the amoeba themselves. Okay. What sample is it that we're looking at? This is a sample from the River Seine in Paris. Again, some of you might have joined a little bit later. I'm always showing at the beginning, I'm showing you the place where I took the sample. And I'm also, of course, you can watch the full video later on when I'm not live anymore, then it's available on YouTube. But I will also make, and I'm working on videos for my main YouTube channel where I'm showing you the same thing, only a little bit shorter. Okay. So, okay, here, this is again, I'm following around now the flagellate. You see there's this long, thin extension, the flagellum. There is a question here. Do you know what's the pulsing dot on the rotifer? I think you're referring to the mouth. I think it's called the mastax of the rotifer. Is this in the middle of the rotifer? Now it's gone. Here is again the vorticella. This is the mouth usually. I think that's what you mean with the pulsating dot. The rotifer, they have some cilia, like a crown, moving the water around, but the actual mouth is further down. So I don't know if I'm able to see it here because the guy is moving so quickly. Up, now it detached. Okay. So I don't know where it is now. So the mouth of the rotifer is actually kind of in the middle of the... look at this oval thing here. This could be right next to the sand grain, to the right of the sand grain. This is oval egg-shaped structure. That could be a test of an amoeba. The test is a shell. But there's a little bit of movement in here on the bottom. I don't know what this is. It's kind of small. So where are all of them right now? Sometimes what can happen is that some of the microorganisms, if you observe them for some time under the microscope, they will actually start to move to the side of the cover glass because this is the place where there is more oxygen. You can see this with bacteria as well. Yeah. Yep. That's another vorticella. Did you see it contract? Okay. Yeah, it's hidden away now. Yeah. Here, I don't know if you're able to. Yeah, the rotifer is again extending. Not the rotifer, the vorticella is extend again. You know what? Here is the rotifer, and maybe you're able to see the pulsating mouth. So yeah, I'm gonna go down. It's 40 times right now, so it's a little bit. The magnification is a little bit. Need to adjust the brightness again. Close this down a little bit here. Yeah, I'm at the edge of the cover glass. So this sample was actually, much better, but maybe not necessarily only because there were many organisms present in the first place, but also because many of them survived. Yeah, look at this. How many worms there are. Yeah. So I'm going to show you the same specimen again, but slightly different because a short recommendation. This is what I've done 2 days ago. I'm going to show you the desk again. Look, this is a plastic box, and what I've got in here is I 3D printed myself a rack like this. It's like a small table, and then there's water on the bottom. So if you basically put slides on here and a lid on top, then the water on the slide is not able to evaporate, and you basically can almost keep it indefinitely. Yeah. So you prepare a slide and a wet mount, and you want to observe it over and over and over again. And you do not want the water to evaporate. Well, then you simply put it into a container like this, and then you put the lid on top, and then sometimes there is actually some water condensing on the slide as well. So I made this 2 days ago. Okay, same sample, and let's have a look at this, and let's see if the organisms survive. Of course they did. So otherwise I wouldn't be showing this to you. Yeah, so that's the one sample that I made now. Look at this. Isn't this crazy? Yeah. Okay, and now I'm going to put in the other one. And this has been on the slide now for 2 days. Yeah. And there is still some movement going on. And I was able to see also some of the worms here. Here they are. Okay. So basically, I wanted to make it clear here that it is possible to also store temporary slides, like wet mounts. It's possible to store them as well if you put them into a container, which is moist, so-called a moist chamber. Here again, that's another one. This is not the flagellate here, but that was another one that I It's too dark again. Yeah, still another one here. Mm. Yeah. Yep, lots of stuff going on here. So let's turn up the light again a little bit. Yeah, again, this here is a sample, yeah, that has been on the slide for two days now. Okay, and I did not add any water. Simply it was kept moist by the moist chamber. So you see that, yeah, this is also a way to kind of preserve slides over a longer time. Look, there are other single-celled microorganisms over here floating around, and here all the way on the left, gastrotrick on the top, and all the way on the left is again are those flagellates that I showed you. By the way, in case you're interested, maybe that is a nice possibility now. Just going to also show you, I also have a 40 times phase contrast objective here on place. I have to turn this up. And let's move this out here. Just a second. Yep. So this is now phase contrast, and now you're able to see the flagellum much better. The flagellum is this long line that extends from the cell, and it's beating and it's pulling the cell along. Yeah. The question again, is the water under the cover glass in the moist chamber replaced somehow? No, I did not replace it. What happens to the oxygen under the cover glass during the stay in the moist chamber? And that is the interesting thing, because the cover glass, there is enough oxygen. That's the interesting thing, because the amount of water is relatively small and there are four corners of the cover glass. Oxygen is able to diffuse into or beneath the cover glass. And this is actually one of the reasons, I think, why the water from this Seine River, which contained actually only a very little amount of water, actually stored better than the ones which contained more water. Yeah, so I'm going to show you the other one over here. Yeah, this is another one that I took, and this started really to decompose quite quickly. And I think one of the reasons could be is that it started to become anaerobic. Yeah, so I actually thought that, well, let's take along a lot of water, a lot of sample, because this kind of stabilizes everything. Because I used to have an aquarium, and usually aquariums with a lot of water are more stable. Okay, there are less temperature fluctuations and so on. However, I found out that if you take along a lot of water and you seal it off with a cap, then it's not so stable, obviously, because oxygen exchange cannot take place. And especially if the tube is very thin, like this one over here, and there's a lot of water in there, then less oxygen is able to reach down. And in the case of a microscope slide, like I have over here, there is very little water in there. And there is, always a possibility for oxygen to diffuse in from the side. And I always kept it at a relatively bright place, not warm, because this causes overheating. But if it's bright, then the algae and the diatoms that are in there, they will also start to produce oxygen. And those microorganisms that move around so much, they need a lot of oxygen, okay? Yeah. As somebody is an expert here, the flagellate belongs to a genus of algae called Peronema. Okay, and so better surface to volume ratio. Yes, exactly. Yeah, at least that's my hypothesis. Yeah, because I've taken along some water samples that looked very great at the beginning. A lot of water and a lot of algae, and I capped it off. And I was kind of thinking, okay, the algae, they'll take care of the oxygen production, and they did, but only up to a point. Yeah. And then it started to decompose. Yeah? I mean, if it were that easy to make a closed system, then that would be great. I mean, then you'd have a so-called a mesocosm, where essentially it's a completely self-contained system, but that's very difficult to do. Yeah. Let's think of an experiment. Yeah, exactly. That maybe indeed that's something that I could actually try to do. Is start off with different water samples, and then have different amounts of water, and maybe cap off some of them and others not. Yeah. Phase contrast looks amazing. Could it show you all the DIC? Well, I've been using DIC pretty much all the time. So for my 4x objective does not have DIC. In case you're kind of wondering, you might see that every time when I change the magnification, it also changes the light intensity. Sometimes I have to readjust this. So I don't know, I'm just going to show you again a little bit of DIC. I have to rotate the filter here on the bottom as well. And yeah, I'm going to turn up the brightness. And there is also a prism that I can change here. Yeah, and when you rotate this, you can also make adjustments. And basically, you might not notice it here so much, but DIC does give you a slightly more three-dimensional view. But I'm going to show you now how it would look like under bright field by simply removing the polarization filter on the bottom. Okay, it looks more flat. It depends really on the specimen, but that's now regular bright field. Yeah. So I would say that these this sample is not such a good one maybe to demonstrate DIC. Here there is a rotifer. Okay, so that's now regular bright field. And second, DIC requires a lot of light. And now this is now it looks a little bit more like like it's standing out a little bit. And now you might we also see this pulsating dot, yeah, in the middle. That's the mouth, the mastax as it's called. Yeah. Phase contrast is here iolosoma at the top. Phase contrast is good for very thin specimens, very small specimens, also bacteria, for example, or you could see the flagellum quite nicely. But phase contrast is not really very effective or very good of thicker specimens. Okay, it doesn't look quite as good. So as you can see, every contrasting technique has its own advantages. You have, for example, here, let's have a look at this flagellate again, okay? The one that's at the top, okay? This is now, it's disappearing a little bit behind the other, okay? This is now in DIC. This is in bright field. It's losing focus a little bit, okay? And when I switch over to phase contrast very quickly, I have to turn up the light again, and I lost it again. Yeah, then you're able to see, here it is. Okay, then all of a sudden you see the very thin structure, the flagellum, this long line that extends, yeah, is now much more visible in phase contrast. Yeah. So you see that you cannot say that one contrasting technique is better than the other. It really depends a little bit on what you want to look at. In case in my microscope I only have 40 times phase contrast connected. Yeah. So you see phase contrast good with very thin objects. A second one. Yeah. And here very nicely visible the flagellum. And if I basically switch over to the other, okay, very quickly I have to change the filter setting as well to refocus. Yeah. Then other things become visible again. You see, that's one of the problems at higher magnification, that you tend to lose the things quite quickly. Yeah. But maybe we just use this this debris that we have over here. Just look at it, then you might see that it looks a little bit three-dimensional, or maybe the algae looks a little bit, like some parts are standing out, like there's light coming from the side. I'm going to switch over now to bright field. Yeah, and it looks more flat. Okay. And here this debris also looks a little, here it is again, the flagellate. Yeah. As you can see that, and again DIC, I have to increase the light intensity, and here you see it quite well that it looks like there is light coming from the top corner, from the top left, and that is basically gives it a somewhat of a three-dimensional view. However, then that's also important, it does not mean that the organism actually has this shape, or three-dimensional shape. Yeah. and the flagellum, this long line that extends, yeah, you do not see quite as well as with phase contrast, for example. Yeah. What scope is that? It looks nice in turn being expensive. Yeah, it's an Olympus microscope. And when you buy microscopes from these large companies, usually what they do is you cannot just say, I want to buy this microscope. They're going to ask you, okay, what objectives do you want? What filters do you want? What condenser do you want? Basically, they put it together based on your own needs. Normally, the lower cost microscopes that you buy, they come finished out of the box. With those brand microscopes, they don't sell them out of the box. You basically talk to a salesperson and you discuss with him or her what your needs are, and then basically they'll make you an offer based on your requirements, and then you have basically you have to know what you want really. Yeah. And if you need some replacement parts, you just call them up and they'll send you whatever replacement part that you want to have. Yeah. So I still actually have a sample that I want to, I've been already talking 3/4 of an hour, but there is still another sample that I want to show you. And I'm going to, just a second. I also made a permanent mount of this. So maybe I'm just for comparison, also going to show you the permanent mount, which basically I also made just a few hours after I collected the sample. And this is how it looks like. The same, of course, no movement, obviously. I mean, everything was in glue. And you can see that there were plenty of algae here. And of course, as the glue dries, this might actually kill, not only might, it will kill some microorganisms, or all of them. But that wouldn't be the problem, but sometimes the glue also kind of dissolves them, and then you cannot see them anymore because their structure is not preserved. So this is a little bit of a problem. It would be kind of nice to see now, okay, some of the worms or whatever, yeah. But sometimes, yeah, it's not preserved, yeah. So I had actually problems finding the same cells as I basically found before. Yeah. So again, this is the same specimen in a permanent mount. So generally what you do is you use a water-based mounting media. In this case I use the glue for water organisms, because if you use solvent-based mounting media, then it can quite be possible that many of the structures you're not able to see because they're dissolved by the solvent. So I have a last sample that I want to show you. Not quite as exciting as the one that I had right now, and this one was a little bit of a frustrating one for me because I really had high expectations, and then I collected a lot of it. I'm just going to show you now the desk again. I took it out and I poured it over in a jar, but originally I had it in a tube like this here, okay, filled all the way to the top. Lots of algae that are collected, and then I opened it and it started to smell sulfury. And I said, oh gosh, no, decomposition started. And just a few days before, I could see bubbles of oxygen in there. So I said, a lot of oxygen production, it's living and thriving. And then within a very short time, it started to decompose because evidently bacteria, they start to grow exponentially, and they've removed all of the oxygen and or most of it. And then it smells bad. Not a good sign. Okay, this basically means that decomposition has taken place. However, if we're lucky, we're still able to see a few interesting things here. Not all of the life was killed by the lack of oxygen, and and it starts to turn turbid. If you look through it and it's not clear anymore, if the water is kind of turbid, look at the green screen. It's also removing, it's also removing the sample here because the sample is also green. So this is a little bit, here you see it. This this is kind of a thing that if it looks turbid, then this basically means that there is some bacterial growth going on here. Okay, so let's give it again a try. I'm going to remove the excess water here, and yeah, there are again slightly different living things in there. Now I'm looking for my cover glass. Here they are. And cover glass goes on top as always. And let's have a look. No, I want to show you first where I collected it. Okay, I'm gonna show you the video first. So this is basically what I did. So on the west of Paris, there's this nice, beautiful park, and yeah, I checked in with a boat. Yeah, this is the third sample. That's the third sample here. This is the third sample. Which I collected in this tube where I had so high expectations, and then I basically kept it closed, and then decomposition started. That's in Paris. There is a park on the west side of Paris with a huge pond, and yeah, I took a little boat and spent an hour there, and I added, it's great, a lot of water. Yeah, and this is basically what I collected, and I put a cap on top, and said, great, this is gonna last now. You see, almost filled all the way to the top. Yeah, and then it started to decompose. Yeah, so this is okay. Yeah, the video starts again. Yeah, so this is a little bit of a disappointment, but yeah, I was still able to see some of the things here. So we're gonna have a look here, microscope view again, and this is now the one that I took at the pond. And maybe I've taken a little too much water here. And you see a lot of, I don't know, maybe smaller ciliates and so on that are there, more light. Yeah, and the algae, they kind of look broken down and decomposed. And you're going to say, well, there is some stuff floating around, some movement going on. And I think, yeah, what happened here is as it started to decompose, many of the other living things, they start to die, obviously, and then other microorganisms start to become more dominant. And those little dots that are floating around, they seem to feel just happy and fine here. And they basically are able, they have adapted to this environment and said, it's fine, great. But what about, I don't know, water fleas or other, yeah, large ones? I could not see. Yeah. Yeah, as a matter of fact, I did find several diatoms here. So I'm right now using my 10 times objective, but I was able to find here, like over here, some colonial algae, and some of them looked quite nice. See, it is not enough light here. And if you look at this, yeah, in the corner, in the top left corner, looks a little bit like a star. Pediastrum, that is the name of this, yeah, algae. Again, a question, yet another question. If I'm able to isolate a single-celled organism with a pipette, what does it take to start a culture? I'm interested in doing this with... Okay, I don't know those, the left yeah, but is a bacteria a single-celled organism? Yes. First of all, chances are pretty good that with a pipette you might not be able to only take one, but sometimes in many cases you're going to take, without knowing it, also a lot of bacteria along, and it might be that the bacteria start to reproduce in that culture as well. Okay, yeah, yeah. So this is a thing. It depends very much. If you want to isolate a single bacteria, you use Petri dishes and agar plates to do that. That's a standardized microbiological process. And here you have to basically isolate individual microorganisms. Usually it is possible to get them with a pipette, but you use a so-called a micromanipulator. So what you do is you have a very, very thin pipette, which you have to make yourself. I've actually tried to do this. You need a glass pipette. This is plastic, okay? But what you do is you heat the glass pipette over a glass flame, and then you pull it apart. And this gives a makes a very thin capillary, and then you can put it in there and you can actually isolate individual cells like that. Yeah? But in order to to get a pure culture, you need to transfer this into a culture medium without actually transferring any bacteria or any other microorganisms. And this means that you have to work in a very sterile environment. Yeah? Or the medium is in such a way that other organisms are not able to grow. Yeah? But yeah, so these are colonial algae I found here. Yeah. And I've found several of those. And you see, I don't know, these seem to be like ciliates floating around, I guess, or flagellates, doesn't matter. But they seem to be pretty much quite the same. And it could be that, as I mentioned, that they were not present to that extent at the beginning, but that they simply multiplied because they simply were able to tolerate the low oxygen concentration. So again, my hypothesis. Yeah, those long stick-like structures, these are diatoms, also to be expected. Yeah. Yeah. And yeah. And also the algae, they don't look quite as nice as I kind of expected them, because when I collected them, yeah, you could actually see the individual strings, the filaments of the algae, but right now it's just a mess. Yeah. So this is a sign that some decomposition, or quite a bit of decomposition, has already started. Yeah. I was able to yesterday isolate an ostracod, which is also a crustacean, which somehow survived. And occasionally also yeah, some nematode worms. This round structure seems to be the shell, is so-called a test of an amoeba. Tests are, that's the name that you give to the shells of amoeba. Yeah, so this is a little bit the mistake that I made, okay? I did not keep the tube open, but because I was kind of worried that I'm going to spill it, so I always kept it closed, and I kind of trusted that the algae are going to produce the oxygen, so I kept it also in a bright environment. And this worked for several days fine. But then all of a sudden it very rapidly started to break down, and yeah, and I did not see that, and I simply reacted too slowly. Yeah. Yeah. So I'm going to now show you here as a last thing. I'm going to show you. Yeah, we learned something from your mistake. Yep. I also learned something, but at least I can pass on something right now. Yeah. But I'm going to now show you also the permanent mount that I made again when the sample was fresh, and I'm going to show you now how it originally looked like. Okay, just remember the mess right now, and now almost 2 weeks ago, how the sample looked like almost 2 weeks ago. Look at this. Here you see now the filaments of the algae much more well. Same sample. Yeah. Can we see some bacteria in the slide? Yes, I'm gonna, okay. If you wanna see bacteria, I'm gonna show you some bacteria under phase contrast, okay? But I just wanna show you this here first, because here, let's go down with the magnification. I was able to see some water fleas, of course not moving anymore, because yeah, this is in a permanent mount, and I don't know if I'm able to find them. Yeah, but obviously you see, look, here at least the algae are still intact. Yeah. I don't know where I found 1 or 2 water fleas in here as well, preserved in the glue. Here it is. Here's one of them. No, it's a water flea. Yeah, some bubbles here. Yeah. If they're moving around, then that's of course very nice to see, and this was kind of the hope. Okay, that I'm able to show them to you. But right now, I just can demonstrate that they were present in the original sample, and this is how the sample looked like when it was fresh. So the glue was not able to completely destroy the shape of the water flea. So yeah, you want to see some bacteria. Okay, so I'm going to show you some bacteria as well. So I'm going to use a slide which I made. I don't know which one. Maybe I think the one from the beginning was okay. I'm just going to, I don't know which one. It doesn't really matter, I guess, because all of them contain bacteria. But the bacteria are otherwise quite difficult to see. I'm gonna go up with a, I don't remember which one was this again. Ah, yeah, this was the one of the, this was actually the good one, from the Sen, which contains probably less bacteria because all of those microbes, they eat, and the worms, they eat the bacteria. So let's maybe use the other one here. I think this is the one, if I remember correctly, from the fountain. Sliding diatom. And we have to go up with the magnification if you want to see bacteria. I've got to go up with light intensity, and I don't know if you're able to see this. No, I hope that the video is going to be good enough. Let's go a little bit. No, not yet. This is not phase contrast. Let me quickly check the microscope here. Just a second. Yeah, they are difficult to see because of the low contrast. So let me try the following. I'm going to go directly to phase, and then let's do it like this. Let's open the condenser, and no, this is not a good one for bacteria. Seriously, where are the others? I'm going to, you know what? I'm going to make a fresh sample. I'm going to try to take one now from here. This is the one which really decomposed a lot. Okay, and let me see if this actually works better. And I'm going to take a small amount here. And I expect a lot of bacteria in here. But you know how the demonstration effect is. If you want to observe bacteria, you need to really make sure that there is very little water on here. So microscope view. So 40 times, objective 10 times. No? Ah, that's why. Wrong setting of the filter. So 20 times. 40 times. And a little bit more. They're not very interesting. Many of those dots are bacteria. I was hoping actually for some more interesting ones, like spiral shaped ones, which I was also able to see. But I'm going to, this is now DIC, okay? Ah, see, there is one, maybe one over here, swimming around. Yeah. But I'm going to now switch over to phase contrast. And let's hope that we're able to see bacteria better. I need to open this a little bit here. I need more light, and no, that's the wrong one here. And let's do it like this. Yeah, I've also, yeah. Yeah, okay. Do you see those corkscrew about my eye level on the other side? Yeah, eye level, there's this small spiral-shaped structure almost in the center. That's of, yeah, these spiral-shaped bacteria. Okay, yeah. Yeah, so I've seen, actually I had a more interesting specimen as well. But at least you are now able to see that bacteria are quite small, yeah, and yeah, even this sample is not the nicest one. but it demonstrates a little bit the importance of phase contrast, especially when you want to observe bacteria. Yeah. Because they appear dark on a brighter background. Yeah. And some of them move around as well. Generally, many, I mean, I'm a teacher as well. So when I teach my students microscopy, the first thing is they want to see, okay, I want to see bacteria. Yeah, I can show it to them, but generally I do not consider them so very interesting. Yeah? They're pretty small, difficult to see. To see them somewhat properly, like here, you need phase contrast. I think these could be clusters of bacteria here, right? And on the bright field, it's also possible to see them, but you need to really close the condenser quite a bit. So there are more interesting, I would say, specimens than bacteria. But you can quite often see that, especially in those specimens that start to decompose, there are more of those bacteria. So this is kind of, yeah, I've just a few days ago, I've actually seen better examples, but yeah, these are all of these dots are bacteria. Okay, so it's almost, as you can see, a little bit of luck is also needed too. Yeah. So let's go back now to DIC. How this would look like in DIC. Yeah, you see that now the bacteria, or those dots, they look like they're bright on one side and darker on the other side. Yeah. And in bright field, like most people have, okay, I need to adjust this again, and I need to close the condenser. Yeah, it would look a little bit like this. They're more difficult to see a little bit. Yeah, yeah. So basically, what I always tell people when they first get a microscope, they say, okay, can I see bacteria? I say, yeah, you can, but honestly, there are way more interesting specimens around. Yeah, larger ones and easier to see. Yeah? And this is the reason why. I think also in biology education and microscopy education, we have to get away from thinking that you have to magnify a lot to see a lot. No, the most interesting things can actually be observed at low magnification. So a similar thing also in astronomy. People who are watching stars, there's some people, companies, they sell telescopes and saying, yeah, it magnifies more and you're able to see more. Not necessarily. So yeah, so 20 times. So this is basically, yeah. Generally, I have to say the following. Yeah, let's go back here as a final comment, because I've been already doing over 1 hour. When you look at a specimen like this, which was actually totally green at the beginning, okay, and now it looks yucky and smells bad. Which bacteria are in here? I have no idea. Really, I have no idea, and generally. It's not advised to actually have stuff like this. You got to be careful. If you spill it around, I have a very high concentration of bacteria somewhere, which I don't know what they are. I don't assume that they're automatically dangerous, but you don't know. And if you're inhaling this and if you have aerosols, basic hygienics is important. So I have less concern with those specimens, which I had over here, like the water from the river. Okay. Where it's clear it's green, so there are lots of larger organisms in there that water fleas, maybe certain worms, they feed on the bacteria and they keep the bacterial concentration low. So I have less concern with those, right? I mean, when we swim in a healthy pond, okay, yeah, then there will be a lot of larger organisms in there that keep the bacterial concentration low. But as soon as you start to add nutrients, like if you over fertilize a pond, or if you start introducing nutrients somehow into the system, this could be, I don't know, dead animals, for example. Yeah, they're like nutrients, and bacteria start to grow. And then it's kind of getting, it starts to smell bad, and it can be more of a health issue. I'm not saying that there are no bacteria in there, but at least it's probably less to be concerned about than a sample like this one over here, which is really spoiled. I mean, it's like decomposing food, for example, rotting food. It's also not healthy, right? So this is basically what I'm saying it's important to kind of keep the oxygen concentration high somehow. And this was my basic mistake, which made me a little bit upset with myself, that it should happen to me. Yeah, I simply had too much of a trust. Yeah, I said, just fill it up. Plenty of algae, put it into light, it'll take care. And then oxygen was active, and I could see all of the bubbles. Great, and it's going to last for 2 weeks now. And then very quickly, within a very short time, I was not checking the tube frequently. Within a very short time, it started to swap over and it started to decompose. Yeah? So keep your tubes open. Yeah, and this is also one of the reasons why I immediately transferred it again into a jar like this with a large surface area for oxygen exchange to take place. And carbon dioxide from the air can also go into the water for the algae to grow, to do photosynthesis, because these plants, they need carbon dioxide, which they also get from the air. Yeah. So this is actually a much better thing, and now I kind of almost completely destroyed it. Okay, so that's why I also learned quite a bit, using these experiments. Okay, so there are some other comments here. So do you think it's okay to wash fresh water slides with the sink? Should that be safe? I'm somewhat concerned about that. I'll be very honest with you. Look, the water that I've collected from the river or from a pond, I mean, if the pond is clean, people swimming in this. I mean, there are. So it's not a problem, of course. Yeah, because this is the stuff that I'm in contact with anyway. Yeah. When I go into a pond like I did the day before yesterday, I went swimming, and you feel that the rocks are slimy because there are algae growing on top of it, and you swim around, and some algae are starting to pass by, and then some green stuff is floating around. And that's a completely normal part, because ponds that are made available for swimming, they check the water quality anyway. So you see, I would only swim in those places that are checked anyway. Yeah. So this is a completely normal part. But honestly, if decomposition starts, if it starts to smell foul, like sulfury and so on, then this is a sign that bacterial growth really started to increase. And then I would be careful. So what I would, for example, do is with those tubes here. What I'm going to do is I'm going to dump it into the toilet as it is, and then I'm going to rinse it out, of course, carefully, and then, or I add some alcohol in here, or I simply throw the whole tube away. Yeah, but I mean, I would not, especially with this sample over here, which is decomposing so much, I'd be careful not spraying it around. I don't automatically assume that it's dangerous, but I don't really know what I'm inhaling. And the bacterial concentration in those samples is really high. Yeah, so it's not necessary. Yeah? So generally, what they're advising is also during when I did my microbiological training, and I was also working with unknown bacteria. This was part of my research. And this was already an elevated safety category. Yeah? But the same thing happens when you, for example, let food spoil. I mean, if you leave I don't know, the other day I had a cucumber in my refrigerator, which I forgot about. It started to get bad, and there were bacterial colonies growing on the cucumber. You don't want to mess around with that as well, right? Yeah. So what is it? So what I'm going to do with this here is because those tubes are cheap, and I'm just going to throw it away as it is. But generally, also, for example, if you have biological, when we were collecting, for example, potato peels and apple peels, all the biological waste, it's being composted. You also got to be careful with the compost. There are lots of microorganisms growing there as well, and inhaling this stuff is also not the healthiest thing. Yeah, so you see, I recommend that you have some kind of a common sense type of approach. You don't automatically have to assume it's dangerous, but at the same time, it's not necessary to be a little bit careless. Okay. Yeah, so this was a little bit, why I'm elaborating on this is because I've received this question already several times, and especially if you go online and if you Google a so-called a hay infusion. This is basically where you take dried grass and you put some dried grass with some water and then you let it stand for some time. This is I don't know, when they started out in biology, microbiology a few hundred years ago, this is what they called, they made this a lot, those hay infusions, and then they used the bacteria and whatever organisms they found, they put under the microscope. That's why they used to call them infusoria. The first microbes that they found, like the ciliates, they called them infusoria because they were from a hay infusion. But it's generally not recommended to make those. At least I don't recommend to make those hay infusions with students, for example, because then you have a whole jar which basically contains decomposing plant material, and it's not necessary. Yeah, and you're spilling something, and then you have got a lot of bacteria in there. It's just like spoiled food. I mean, you don't want to mess around with spoiled food either, right? So that's why I'm saying use common sense. Hygienics here. Yeah. Have you ever put parts of a puffball fungus under the scope? Speaking of lots of stuff, not a puffball fungus, but yes, I've put a fungus of mold. For example, oranges or lemons sometimes get moldy, and if you put this stuff under the microscope, you see a lot of spores. And in my Microbehunter main channel, several months ago, if you just search for spores, I made several videos with spores, and the number is really high. And of course, inhaling this stuff is also not the healthiest thing. So at least for those people who are maybe allergic. Some people, when they inhale spores, they get asthma and so on. But it depends a little bit on the type of spores that you have in there. Yeah. So, but oh my gosh, I've been talking now again. It's 1 hour and 18 minutes now that I've been doing this live stream now. If you're interested in stuff like this, I'll be posting videos on my main channel about the things that I collected over the holidays. Yeah, and there's another one here. Hey Oliver, I just came home from work, your live stream. I actually have my biology exam on Wednesday for school, grade 11 in Ontario, Canada. I wish you all good luck for your biology exam. Okay, yeah. Basically, I'm, as a biology teacher, also, of course, it's also part of my duty to give biology exams. Okay. What's the easiest sample or infusion for education? You know what? What I would suggest that you do. Okay, I think that's an important one. You want to, if you want to show ciliates, let's say paramecia, then do the following. You take a water sample and you crush a wheat grain. Yeah, for whatever reason, wheat grains kind of work best. You take a wheat grain and you crush it between 2 spoons and you put it into the water sample, just 1 grain. And you wait for about 3 or 4 or 5 days, and then you put a little bit of the wheat grain under the microscope. It turns slimy, and if you're lucky, then it will contain a lot of paramecia. Okay, it's a standard way of grow. What's going to happen is, yes, bacteria are going to grow on the wheat grain, and then the paramecia will feed on the bacteria. So you're establishing a small food chain. Yeah, but I would always, of course, not deliberately grow bacteria by decomposing, let's say, hay infusions and so on. Because if you put a hay infusion with water and some dried grass, the paramecia are not going to appear because they cannot be found in the grass. So what you have is you have a whole bunch of bacteria growing there. So basically, clean water samples are usually the best. If you've got an aquarium, make an aquarium. Okay, if you want to do something with students and so on, make an aquarium. This is actually, because the water in the aquarium has to be relatively clean anyway. Yeah. Okay. Wow. Genetics is killing me a lot, but I love it too. I want to do something with the future for teaching. Thank you. Yep. I had a recent biology project and I was able to use my microscope to show microorganisms. It was very fun. Great. Just wondering if you have some colored filters templated we can download and put Reinbook filters. No, I don't have them for download, but I would actually say it might not make sense because it might be different for different microscopes. Okay, so I think there might not be a one-size-fits-all solution here, and you have to maybe make your own Reinberg filters. I made separate videos on this as well, but I think you have to adapt it a little bit to the microscope that you have. Okay. I did the crushed grain trick. After a week, it started to smell like a sewer. Then it went anaerobic. Okay, sewer is not good. Huh? And you have to start off with water which is relatively which contains the paramecia. Okay, that's important. So you probably don't want to use just water, but maybe some algae that contain, or some material from the sediment that actually contains those ciliates. Okay. Yeah, okay, I have a 350B. Yeah, that's a fine microscope. I will try to have any food. Okay. So any good like to buy. Okay, I would like to, I think I'm just going to leave it at that. Thank you for your comments. This video will be available, of course, also for those of you who join in late. You can, of course, watch the whole video then as well. I'm going to leave it at that for right now. Yeah, to attract the T4 similar bacteriophages, questions keep on popping up here. To grow medium, or do you have to buy them from a biosupplier? Plaques. Honestly, you want to do T4 bacteriophages. Yeah, they can be found there where there are bacteria. There are bacteriophages, and of course you can get them from water samples and so on. But honestly, nah, don't grow bacteria at home using Petri dishes unless you know what you're doing. Okay, that's my recommendation. Unless you know what you're doing. There are so many other things that are also interesting relating to microscopy. So I think I'm really going to force myself to stop now. Yeah, if everything works out, I'd like to do another one maybe next week. Maybe I'm going to show you some specimens that are collected from the North Sea, from the Atlantic. Yeah, but for right now, I'm going to wish you all the best, okay? And see you around next time, and happy microbe hunting as always, and bye-bye everyone. Bye-bye.